So use or applications are multiple. Applications of MOABs. So applications of MOABs are multiple. Okay. So how they are, what are their major applications? Let us say the major applications would be, okay, the major applications of monoclonal antibodies would be, first and foremost is for in vitro diagnosis. Okay, why? For in vitro diagnosis. So one of the very important applications of monoclonal antibodies is in vitro diagnosis. Then what else? Then is therapy. Okay, which is very important. Now why these are important for therapy as well as in vitro diagnosis? Because monoclonal antibodies essentially are highly specific. Why? You may ask why? Because these are highly specific, high specificity, okay, and high selectivity. Both are there. So what happens? For example, in your test tube, or let us say for there is a Petri plate. Now in this Petri plate there are multiple different, what do you call, say kind of colonies. Okay, so this is one colony, this is one more colony, this is one more colony of different organisms. Okay, now you want to know, but all of them are looking exactly the same. Okay, now you have to differentiate whether these are different or not. So what you will do, you will use a monoclonal antibody against colony 1, MOAB against colony 1. Okay, and then you will pour it on this plate, and eventually you will see that antigen antibody interaction is taking place only against a particular colony, not against everybody else. So you will find it out that there must be a different species of organism here. So this is, you will term as organism 1. Okay, now consider that you are having the monoclonal antibody against coliforms. So let us say anti-coliform MOAB. Okay, so what will happen? You will be certain that in your colony or in your Petri plate, the organism 1 is coliform. Okay, coliform. Okay, now the organism 2 is Vibrio. This is a different organism, Vibrio. Because why? Because it has reacted with anti-Vibrio antibody. Okay, this kind of technique is also useful in order to identify different species. You are having a battery of monoclonal antibody against it. The other example is blood grouping. Okay, very common example. In blood grouping, what will happen? You will use a particular antibody, let's say anti-DMOAB, monoclonal antibody. Okay, so what it will do? It will only identify a particular antigen which is known as RH. Okay, now if RH is present on this particular, what do you call, RBC, okay, now this is RH antigen. Okay, if RH antigen is there, then only anti-D antibody will make it into a precipitate, and you will understand that your blood group is positive. Okay, so there will be a precipitate, and eventually you will understand that the blood group is RH positive. Similarly for rest of the other blood groups as well. Okay, for all different blood groups we are having NTA, all are monoclonal antibodies. Okay, NTA anti-A, MOAB, then anti-B, MOAB. These are all monoclonal antibodies. Okay. Now I'll come to the last point, that is for the therapy. Now this is for in vitro diagnosis. Okay. There are plenty. For example, consider this: you are having a kit which is called hepatitis B surface antigen kit. Okay. Consider that you are having a kit which is called HBsAg kit. Okay. Now in this kit, what is there? There is a particular sample well, okay, where you will add serum, and there is a window. Okay. Now in this window there is a positive control and there is a negative control. Okay. So whenever by a dropper, I won't able to this thing yet. By a dropper you will add serum sample. Okay. Drop by drop. Drop by drop serum sample you will add. Consider that this is your serum sample drops. Okay. So what will happen once the serum sample has been added here? There will be a what? There will be a diffusion. Okay. Because here some channel characteristics will be there. On the surface there will be a particular kind of matrix. Okay. In this there will be a particular kind of matrix, and due to the presence of the matrix there will be a diffusion of the serum, and the serum will start diffusing towards the well, reaction well. Now here what will be there? There will be a anti-HBsAg monoclonal antibody. Okay. Now here there is what? There is a anti-HBsAg MOAB. Okay. So what will happen? If at all the antigen is present here, okay, consider that this is specific antigen for hepatitis B. Okay. So then what will happen? There will be coagulation in this window. Okay. There will be coagulation in this window. Okay. Which will be visible, and a particular color will be precipitated, and there will be a particular spot. On the basis of this, there will be a particular spot here. Okay. There will be a particular spot, and on this basis you will say that in the serum sample, which was again in a test tube, in the serum sample, then you will say that in the serum it is positive for, then what you will say? You will say that this serum sample is positive for hepatitis B. Okay. Positive for hepatitis virus. Okay. Similarly, we are also having what? You all know, pregnancy detection kits, on which we are having antibody against what? Who can tell me? In pregnancy detection kits, HCG, anti-HCG antibody. Okay. Human chorionic gonadotropin antibody. Okay. So what exactly happens? Wherever you are having a monoclonal antibody, you can identify a specific kind of what we call what? Antigen, if it is present in there. For example, in ELISA, I'll come to those points specifically later on. In ELISA also we use what? Monoclonal antibody. Now this is whole exercise of in vitro diagnostic tests where you are using monoclonal antibodies. You are also using monoclonal antibodies for some high-end tests. So I'll mention it. Some high-end tests or very sensitive tests like Western blotting. Okay. Western blotting, we are using this monoclonal antibody. Then we are using monoclonal antibody for fluorescent microscopy. Okay. Then we are also using monoclonal antibody based tagging system for techniques like FSCS, fluorescent